In this video, we're going to be going over the details of antigens. Antigens are large molecules that the body is going to recognize as foreign and is going to attack. Antigens are substances that are going to induce an immune response. And if you look at the term antigen, it means antibody generator. So anti for antigen is antibody, and then gen for antigen is generator. In general, the more complex a molecule is, the greater its antigenicity, or the ability to stimulate an immune response. And antigens are usually proteins of glycoproteins, but they can also be carbohydrates and lipids. Each pathogen can contain hundreds to thousands of different antigens, which include bacterial components such as the cell wall or capsule, as well as proteins of toxins, viruses, fungi, protozoa. Even food can elicit an immune response if your immune system recognizes those proteins or carbohydrates or lipids on food as foreign. And such things as pollen and dust can also contain antigenic particles known as allergens, and whether you're allergic to it or not just depends on whether your immune system recognizes them as foreign. You want to make sure you star this. An epitope is a specific part of an antigen that is recognized by the immune system. Specifically the antibodies, B-cell receptors, and T-cell receptors. Antigen can contain 1 to dozens of epitopes. For example, you're looking at a bacterium. These are antigens that are on the surface of this bacterium. These antigens can contain 1 to many what are called epitopes, and it's the epitope that's going to elicit the immune response. So an antigen can have more than one epitope. For example, if you're looking at this one right here, it has 1, 2, 3, 4, 5, 6, 7, 8 different epitopes. And these epitopes, which are part of the antigen, can elicit immune response. For example, where antibodies combine. If you look at this right here, here's the antigen. This is one of the epitopes of the antigen that can bind to this antibody that has this particular shape on the antigen binding site for this antigen, or epitope. Here's another antibody that its antigen binding site binds to this part of the antigen, which we call this an epitope of this antigen. Here's another antibody that's binding to this epitope of this antigen because it has a particular receptor that matches that epitope. Same way when looking at B cells here, B lymphocytes. These 3 B lymphocytes each have different receptors on their surface that will bind to a specific antigen or the epitope of that antigen. We look at the types of antigens. We have 3 main types. We have exogenous antigens, and these originate from microbes located outside the cells. And they include toxins and other components of microbial cells, such as on their cell walls or membranes, flagella, and pili. Looking at this right here, this is a bacteria. And as I said earlier, these are antigens. Well, this is an exogenous antigen because they're outside the cells of the body, and your immune system can react to these exogenous antigens. Endogenous antigens are antigens that are found within the cytosol, such as those produced by microbes that reproduce inside host cells, such as a virus or intracellular bacteria, or tumor antigens, whether they're benign tumors or malignant tumors or cancer cells. Those antigens are then moved to the surface of the cell and can elicit an immune response. So for example, here's a virus-infected cell, and this virus directs the cell to make proteins that are going to make more of the virus. And these proteins, some of these proteins can be put on the cell surface to elicit an immune response. One of the reasons why these proteins are put on the surface is that when you're looking at a virus-infected cell, like you see here, a host cell, and it's directing the making of viral particles to make more virus, some of those, depending on what type of virus it is, are going to be put on the surface of the cell to be a part of that virus when it buds off. So here we have a virus that's been made. It's going to bud away from that cell, and these proteins that it directed to make are put on the surface here, so when it buds off, it's enveloped by this membrane that has these viral proteins on it. For example, when you're talking about the COVID-19 virus or the SARS-CoV-2, you might be aware that there are these spike proteins that enable this virus to bind onto receptors and be pulled in by your cells. And that's one of the ways it's going to be infecting your cells. We'll talk more about this when we get to viruses. Well, these spike proteins were made in the cell that the virus invaded, or put on the membrane of the cell. So when the virus buds off, we call it an enveloped virus because it takes some of that phospholipid bilayer, and there are these proteins, these spike proteins, for example, that you see here. And these spike proteins can elicit an immune response, or proteins that are on the surface can elicit an immune response. We call those endogenous antigens because they're coming within the cell. Or if you have a cancer cell and it starts to make proteins that are abnormal that are inside the cell, those proteins can be put onto the surface by those MHC1 complexes or MHC1 proteins, and then that can stimulate a cytotoxic T cell to kill that cell or be stimulated by that cell. So lots of interesting information coming up when we get more specifically into those MHC proteins or major histocompatibility proteins. Autoantigens are self-antigens that evoke an immune response, resulting in an autoimmune disorder. So the proteins that are put on the cell surface, for some reason, the immune system targets those and thinks they are foreign proteins and then targets those cells, which elicits an immune response. But it's your own cells. It's a mistake by the immune system to target these self-antigens. Let's test your knowledge. Part 1. Pause the video and select the term that makes a sentence correct. Here are the answers. 2. Pause the video and answer the following questions. Here are the answers. And this concludes the video, and let me know if you have any questions.